innovate.
Good afternoon, everyone in China. Good morning, Dr. Mary Hirsch, Dr. Hans Cleaver, Dr. Emma Rollins. I'm Zhen Jixu. I'm the faculty member at Wesley University. Together with me is Chen Wang, scientific editor from Cell Stem Cell. Along with Cell Press, Wesley University had the great pleasure to present you this webinar on stem cell research through application of organoids for development and disease modeling. Organoids are self-organizing aggregate culture derived from human or mouse tissue that generate cell types and organizational property reflective of a developing organ. One feature that is common to all organoids is that they are generated from pluripotent stem cells or adult stem cells, also known as tissue stem cells, by mimicking human development or organ regeneration in vitro. The analysis of organoid formation can provide valuable insight about the mechanism underlying human development and organ regeneration, highlighting their critical value for basic biological research, in addition to their potential applications in pharmaceutical drug testing and molecular medicine. The broad goal of this webinar series is to promote scientific communication, especially when international travel is still restricted. Our previous webinar have attracted a large audience for each section and focus on topics ranging from tumor microenvironment to metabolic regulation and cancer immunology, featuring world-renowned scientists. Today, we have the pleasure to invite three prestigious scientists in the stem cell research field: Dr. Mary Hirsch from Germany, Dr. Hans Cleaver from Netherlands, and Dr. Emma Rollins from United Kingdom. They will present their lab's exciting discovery on stem cell research using organoids. With this in mind, Chen will introduce our first speaker, Dr. Mary Hirsch.
Thank you, Zhenji. I'm Chen Wang from Cell Stem Cell. It's my great pleasure to introduce our first speaker, Dr. Mary Hirsch. She is a laser Max Planck Research Group leader at the Max Planck Institute of Molecular Cell Biology and Genetics in Dresden, Germany. Dr. Hirsch obtained her PhD at the Center of Genomic Regulation in Barcelona, Spain in 2007, and moved to Netherlands at the Hobrecht Institute for her postdoc. There, she pioneered the generation of organoids, including stomach, liver, and pancreas. Following these studies, she obtained an independent position at the University of Cambridge, where she moved her research focus on the understanding of liver tissue and where she made very important contributions. In 2019, she was awarded to the prestigious Lisa Meitner Award from the Max Planck Society and moved to Germany. Her lab established the first human liver organoids derived from human liver tissue from both healthy and diseased patients. Also, her lab generated the first liver cancer organoid model. For these achievements, she has received several awards, including the Hampden Award for Medical Excellence, the Woman in Cell Science Prize from the British Society, and the Amber Young Investigator Award, or the Biner Prize. So her talk today is Liver Organoids for Human Biology and Disease. Without further delay, let's welcome Dr. Hirsch.
Yeah, thank you so much, Kwang Yang and Zheng Ji, for the invitation to be on this webinar. It's a pleasure. I'll try to share my screen now. It should work like this, and you should soon be able to see my screen if I do it like that. And once I see it myself, yeah, I think it should be okay, and you should be able to see it. I hope so. As Dr. Zheng Ji and Dr. Kwang were mentioning, my lab and myself, since I was a postdoc actually with Hans Cleaver, who is joining us today, and afterwards in my own lab, we've always been interested in developing these organoid systems, and basically my lab focused on liver and pancreas. What I'm going to be talking today is about liver organoids, but I'm going to give you a little bit of introduction on the organoid field, and actually was very nicely summarized by Zhengji, but I will just be a little bit more extensive on that introduction, and then I will focus directly on our work on using organoid models to understand liver biology, in particular liver regeneration, and how we use it to model liver disease. And basically, with this slide, what I want to give you is a kind of organoid historical perspective. And the word per se, organoid, was firstly, in the first citation for the word organoid was done by Cochrane and Smith in this publication of the Canadian Journal of Physiology, where they use it not to define organoids as Zhengji, and I'm going to define in a second, but actually they were using it to define a cystic teratoma that was growing in the genital ridge of a baby, a newborn baby. And that was in 1946. And since then, several groups adapted the word and they started to use it to mention three-dimensional cultures that they were trying to achieve in ex vivo. Prominent work from Mina Biesel, as well as Michalopoulos in the case of mammary gland or hepatocytes in particular. But this is a very different definition. What they were obtaining at that moment was very different type of cultures of what we defined later on as, or I like to call it, the modern era of organoid field, which indeed is when the organoid field has flourished. And it would start in 2008 by José Gizazai, by the development of cortex tissue ex vivo from a mouse embryonic stem cells, and the pioneering work from Hans Clevers and Toshiro Sato, by using adult mouse intestinal tissue to generate mouse intestinal organoids. And from there on, many different other tissues, and this slide is trying to separate pluripotent stem cell-derived organoids in the bottom from adult or fetal-derived organoids in the top. And basically, here I would emphasize again the definition of an organoid, and this is how we define it now, and this is what it makes it different from what was before the development of the cortical tissues from José Gizazai from the mouse. And we define it as a three-dimensional structure that is formed by self-organization of both pluripotent stem cells or fetal or adult tissue stem cells, or even differentiated cells, as I'm going to be showing to you later on. And I'm showing you here two examples. One is the first mouse embryonic stem cell derived organoid. It was not even called organoid back then, by José Gizazai in a cell stem cell paper, Hallmark paper in 2008. And in the bottom part, I'm showing you what I would say is the first human tissue derived organoids. The first mouse human organoids, mouse organoids were from Toshi Sato in the intestine, and we did the first human tissues on liver from when I was still on my transition from being a postdoc in Hans Clevers to become independent. These structures, they have to not only grow by self-organization, but they have to differentiate into the cell types of the tissue, or at least some of them, and they should recapitulate some of the function of the tissue of organ. And many groups have defined what we now consider organoid cultures. And on the rest of my talk, I'm going to just be focusing on tissue-derived organoids. And as I said, the Hallmark paper on tissue-derived organoids is from Toshi Sato in Hans Clevers' lab in Nature in 2009, where leveraging on the knowledge of how adult stem cells grow, that were also identified by Hans Clevers himself and Nick Barker in 2007, leveraging on the knowledge of how these cells, where are they located, how they look, their isolation, and also what are the factors that they receive in the native tissue. Leveraging on this knowledge, Toshi Sato and Hans Clevers were able to isolate these LGR5 positive intestinal stem cells and grow them in a dish. Back then, we even called them stem cell cultures, not organoids. But we very early on realized that they generate these three-dimensional structures that very much resemble in the dish the crypt-like structure of the intestinal epithelium. And from there on, myself and the lab started to look into other organs. We started with the stomach because indeed Nick Barker, his again, himself again had shown that stomach stem cells reside at the very bottom of the stomach gland. And I was able to, by modifying the culture conditions established by Toshi Sato for intestine and adding additional factors that are important in the development of stomach epithelium, we could establish these stomach epithelial organoids that indeed very much recapitulate stomach epithelium in a dish. But then the question came, what about non-proliferative tissues? So the majority of the tissues in our body are indeed in a quiescent state. It would include lung, it would include liver, it would include pancreas. And then we wanted to ask the question, of course, it's very valuable that we can establish intestinal and stomach organoids because this was the first time that primary tissue could be expanded in a dish. But despite being very valuable, we were thinking, at the end, we are just recapitulating in vitro what the tissue is doing. So the tissue knows how to proliferate in vivo, and we have harnessed this knowledge to do it the same in vitro. But what about tissues that don't proliferate? And again, we had to go back in vivo and ask the question, how does the liver know? And where are the cells located when they are proliferating? And one moment where the liver proliferates is up and damage. And actually, the pancreas also proliferates up and damage. And by identifying the cells and knowing a little bit where were the signaling factors involved, we could generate the first liver and pancreas organoids from mouse tissue. And this was when I was still a postdoc, and I'm very honored to say that, when I was still a postdoc in Hans Klever's lab. So the rest of the talk, I'm just going to be focusing on liver and how we've used and harnessed the knowledge of growing these organoids from the liver to investigate mechanisms of liver regeneration. And just very briefly for the audience that is not familiar with the liver, because I'll be using some of these words, the liver is formed by hepatocytes, ductal cells, and also a lot of stromal cells. And they are not just, they are making a very beautiful structure called the liver lobule, where the blood flows out in from the end of this hexagonal lobe into inside of the central vein, and the bile acid produced by the hepatocytes that are here in brown is released into the bile canaliculi, which terminates into the bile duct flowing to the opposite direction, and they never intermingle. The liver is very well known to have huge capacity to regenerate up and damage, and this is for all vertebrates studied, and it either regenerates very proficiently, but sometimes it undergoes disease and it either cirrhosis or cancer. And at the cellular level, many groups, including ours, but from Stuart Forbes to Marcus Grompe to Willem Vring, they all were able to identify that actually during the damage repair response, there is two ways the tissue can repair at the cellular level. Either the cell makes itself, so it divides and makes itself, or the cell, especially in chronic damage, acquires a progenitor-like state and now is able to make two fates, itself, but also a total different fate, the hepatocyte. And this, we think on how it works at the molecular level, is going from a quiescent state to a proliferative state to a quiescent state. So what are the mechanisms that control these cell fate changes? And we thought this is where organoids, we thought, could be very useful, because very early on we found that, as I said, they start from one single cell that self-organizes, start dividing and makes these structures that now we can literally break in pieces, and each piece is going to make a new structure. But indeed, what they are doing is they are acquiring, these liver cells are acquiring a self-renewing capacity and now are able to make the two fates, which was mimicking. And now later on, my lab showed, and also the Grompe lab showed, that is a differentiated ductal cell, the one that undergoes a dedifferentiation step and activates a proliferative program to generate the two different cell types, which kind of recapitulates what I just shown you happens in chronic liver damage. So we asked the question, can this system be used to understand concepts of liver regeneration, in particularly the transition from a quiescent non-proliferative state to a proliferative progenitor-like state? And that was the work of Luigi Aloi and Michael Mackay in the lab, who first thing they had to do was to confirm that this was not only a correlation, but that indeed there was a lot of similarity of what happens when you induce in vivo injury to the mouse to what happens in vitro in organoids. Only when we benchmark our organoid system to the in vivo setting, we can use them to investigate mechanism. So what they did, and very briefly, was to obtain the transcriptional changes that occur, transcriptional map that occur during this transition from a quiescent to a proliferative state, and do the same in vitro from the quiescent to the proliferative organoid state. And what they found is that there is a 70% overlap in all the transcriptional changes, which are massive. There is a 70% overlap on these transcriptional changes between the in vivo injury in the mouse and the in vitro mouse organoid formation. And when we go in depth in these transcriptional changes, we have a lot of things happening. And also there is a massive epigenetic reprogramming. More than 55% of epigenetic regulators change dynamically in order to allow one quiescent cell to activate its proliferative progenitor program. And just to cut the very long story short, to give you one hint, that what we found is that actually there is a massive change on the methylation profile of the DNA. So basically, not only in the mouse, but is recapitulated in organoids to go from the transition, exit from quiescent to a proliferative progenitor state requires a massive reprogramming of the DNA methylome, changing many methylcytosines for hydroxymethylcytosines, which is changing the methylome of the DNA from being very restrictive in apposition and not permissive to permissive for transcription. And this was exemplified in this heat map, where many genes, more than 3,000 genes, change their methylation marks, repressive mark for a permissive mark for transcription in green, and they will go back to the original state once the tissue is starting to proliferate. So basically that we've summarized this concept that we were able to obtain thanks to having an in vitro system that was recapitulating in vivo setting. We have summarized this concept by saying that to exit from quiescence to enter in an active proliferative regenerative state, there is a massive changes happening. One of them is that very early on, there is a activation of a protein called TAT1 that changes the methylome pattern from a repressive to a permissive methylome pattern called hydroxymethylation pattern in many genes, including genes involved in regeneration, and that indeed licenses a differentiated quiescent cell to enter regenerative program. So what is the niche? What are the signals that are triggering this change of transitions from a quiescent to a differentiated state, to an undifferentiated proliferative state? To understand this question, what are the signals in vivo that induce these massive changes, we went into the tissue bank. And this was the work of Lucia Cordero and Dobach in the lab. And the first thing they noted is that actually there is a mesenchymal cell here in pink that has these protrusions, these very long protrusions that are touching these ductal cells here in white. And they asked the question, if these cells are to get very close together, they might be a niche for this population. And they started doing damage in experiments in vivo, inducing toxins into the mice and seeing that the liver regenerates. And the first thing they observed is that there is a change on the pattern. And this change was on the following way. So in homeostasis, there was a specific pattern of number of cells of the ductal epithelium in white and the pink mesenchymal population. This was changed during the damage response and reverted back at the regenerative state. They started quantifying the number of cells, and what they observed is that actually the first cells to proliferate are the epithelium, the ductal epithelium. And the mesenchymal cells, the pink cells, they stayed in place. But once we remove the damage and allow the tissue to recover, the pink cells, the mesenchymal population, catches up. And meaning that there was a desynchrony on the time of proliferation. One goes first, and that was implying that the number of contacts between both populations was dynamically changing over this time course during the damage response and the recovery phase. And if we quantify that in terms of ratio between populations, what we see is that there is a drop from a 0.3 to 1 ratio of mesenchymal cell to ductal epithelium to 0.1. And then during the damage phase. And then at the recovery phase, mesenchymal cells catch up, they start proliferating, and they revert back that state. And we had the hypothesis that this change on cell contact was critical for the regulation of the proliferation of the ductal epithelium. We had that as an hypothesis, but of course, asking this question in vivo is very challenging because it means that you need to monitor live in an organ like the liver that is, despite you can do intravital microscopy, the time scale of this experiment was technically very, very impossible, almost very challenging, if not impossible. So we turned back to our organoids. And our organoids, as I just mentioned, they were only epithelia. And as I said at the very beginning, liver is formed by many cell types, epithelial cells and mesenchymal cells. So we went on to try to recapitulate this structure of the ductal epithelium and the mesenchymal cells that we were interested, trying to recapitulate that in vitro. And when you just put the cells together, saying, I'm just doing a culture, what happens is the cells just segregate in the plate. You have mesenchymal cells in the bottom, ductal cells making an V2-4 organoid, but they were not recapitulating that structure. In order to do that, we had to team up with Timo Kohler and Florian Hohlfelder at the biochemistry department of University of Cambridge, where they had generated a microfluidics device to microencapsulate our cells. We were not doing anything. We were just trying that the cells are encapsulated very close together so they can meet. And if we do that by self-organization, they actually generate this beautiful ductal mesenchymal structure where the mesenchymal cells are located at the basal side of the ductal epithelium. And then we started filming these structures, and we found a very interesting paradox. In yellow, one organoid that is touched, one organoid in green that is touched by a red mesenchymal cell. In the same well, next to it, another organoid in green that is next to, but not contacted by, a mesenchymal cell in red. And if we look what happens to them when we film them over time, is that one organoid collapses and the other organoid keeps growing, suggesting that cell contact, it was inhibitory, and paracrine signaling from a close by cell but not contacted was actually promoting growth. To confirm this, we had to devise an experiment where we separated now by force, we forced them to separate by growing mesenchymal cells in one plate, generating conditioned media in the absence of any growth factor, and using this media on top of naive ductal cells. And what we observed is that these cells grow very nicely. And what we also observe is that it's because these mesenchymal cells produce some of the growth factors that are important for the growth of ductal epithelium. And then we did the reverse experiment. We put the two cells together and we started playing with the ratios, because that's the beauty of having an in vitro system that recapitulates your in vivo setting. Now you can start manipulating the system. And we started manipulating the ratio between the populations. And if you remember the numbers I told you, if we just focus on the yellow curve, 0,1 to 1 was the ratio that in vivo is inducing ductal proliferation in the damage phase. And indeed, in the organoids, we see ductal proliferation. When we do the opposite, and we start increasing up to non-physiological levels of mesenchymal populations, actually, we even inhibit to the point of totally abolishing organoid growth if we, for instance, go to a non-physiological 2 mesenchymal cells per 1 ductal cell ratio. But, and more interestingly, the fact of having organoids allowed us to ask the question, is this revertible? Can we revert this phenotype by dumping a lot of growth factors in the media? And the answer is not. So basically, cell contact inhibition in that particular scenario is dominant over growth factor presence, because when we dump a lot of full cocktail of growth factors, the blue curve, we cannot rescue the phenotype. So basically, we summarize these results by saying that in the homeostatic population, in the homeostatic state, the tissue, the ductal epithelium proliferation is kept in check, is the mesenchymal, by the mesenchymal pool. Basically, the mesenchymal cells instruct the ductal epithelium to not proliferate. Then, after damage, there is a stimuli that make these ductal cells to proliferate. We still don't know. We have some hypotheses around that. But the moment that one cell proliferates, the ratio is totally changed, and the number of contacts is changed. And that is a positive feedback loop that allows more ductal cells to proliferate until the moment we remove the damage. And then the mesenchymal cells proliferate, catch up. We know that they actually induce apoptosis to the ductal epithelium, and we believe that this explains the transition from being in a quiescent homeostatic state to a proliferative state and revert back to the quiescent original homeostatic state upon regeneration. And we think that organoids have been a very powerful tool to help us visualize and also to help us investigate this concept. And in the last 2 minutes, I would like to show you how we've been modeling human liver disease. And as I mentioned at the beginning, when I was in my transition from being a postdoc to become independent, we were able to obtain biopsies from human healthy livers. This is someone that dies and donates the organ. And we took 1 cubic centimeter of this biopsy, very tiny, the size of your nail. And we put them in culture, and then these things were starting to grow. We were astonished. There was a long work and a lot of extensive work to try to transfer the technology from the mouse to the human, but by modifying the culture conditions, it was possible, and we could now expand for the first time human primary tissue, in that particular case, human primary liver tissue in a dish. And we showed that they retain this bipotential capacity I showed you before, because now we can ask them to produce some of the functions that the hepatocyte, the other cell type, do, like produce albumin, bile acid, or detoxify from drugs. And then we used and harnessed from this knowledge to develop a model that will recapitulate liver cancer in a dish. And that was the work of Laura Brotier in the lab, who took the approach of taking different liver cancer subtypes, cholangiocarcinoma, mixed subtype, hepatocellular carcinoma, which are very different, because hepatocellular carcinoma, its main origin is hepatocytes. Cholangiocarcinoma, its main origin is cholangiocytes, despite in the mouse has been shown that depending on the mutation, also an hepatocyte can make cholangiocarcinoma and cholangiocyte can make hepatocellular carcinoma. And Laura was able to show that actually this system recapitulates the transcriptional changes of that are present in the patient. They also occur in our organoids. But not only the transcriptional changes, but also the genetic alterations that occur in the patient, they are retained up to 90% in the organoids that we can now expand long-term. And most interesting, they also retain the histological architecture. So basically, we do see in vivo architecture is retained in vitro in the organoids. And very recently in the lab, we've taken advantage of this system to investigate a mechanistic question in liver cancer. And we had observed that 2 Wnt negative regulators, RNF43 and ZNRF3, induce accumulation of lipids and actually induce non-alcoholic fatty liver disease in the mouse. And however, non-alcoholic fatty liver disease is a disease that comes with fibrosis, it comes with lipid accumulation, it comes with inflammation. And we wanted to ask the question, is these 2 negative regulators of the Wnt pathway, when they are mutated, they induce Wnt activation? Is the Wnt activation changing the environment? And that's why you get the fibrosis, that's why you get the inflammation, that's why you get non-alcoholic fatty liver disease. Or is actually a change, an intrinsic change in the cell? Is the activation of Wnt signaling inducing lipid accumulation inside of the hepatocytes? And this is a trigger for the inflammation and a trigger for the fibrosis. So then is when we had to turn back to the organoids, because when we were asking this question in the mouse, we could not detangle what happens from the microenvironment from what happens inside of the cell. So we generated hepatocyte organoids thanks to the work of Hans Clevers and Hui-Li Hu in his lab, who transferred the technology he had developed for cholangiocytes to hepatocytes. We could establish hepatocyte organoids from these mutant mice, and then we depleted the genes in vitro, because that's the beauty of having organoids. You can now do genetic manipulation ex vivo in a primary healthy cell. And that immediately, 5 days later, we took these images, where what you're seeing here is this massive green dots, which are actually lipid droplets that are accumulating in the hepatocytes of the mutant cells, mutant organoids, compared to the parental. These are sister organoids. The mutant came from these wild-type organoids. The wild-type organoids do not they accumulate normal lipid droplets because hepatocytes do this function, but the mutant had a massive increase and significant increase in the number of lipid droplets in the hepatocytes. So that was telling us that the mutation of these two genes also cell intrinsically and cell autonomously causes lipid droplet accumulation in the hepatocytes, and this could be the trigger for the inflammatory reaction and the fibrosis and the NAFLD phenotype that these mice develop. Then we turn to human, because now we had these two genes are mutated in human liver cancer. So we turn to our human liver cancer cohort of organoids, and we checked which ones were mutated in some of these genes, in both or one of the two genes, and we find one, and this is a mixed subtype liver cancer that is mutated in RNF43. And we had a mixed subtype, so the same subtype of liver cancer that was wild type for RNF43. And when we checked lipid droplet accumulation, again, we don't have any stroma. We know that we had an increase in the number of lipid droplets in the mutant cancer organoids compared to the wild type cancer organoids. So that is to give you an example of how using organoid system enabled us to understand a mechanism of a particular disease, in particular liver cancer, how indeed these two genes, the role in liver cancer is to induce intrinsic cell autonomous accumulation of lipid droplets in the cells. And with that, I would like to just summarize what I've been going through with you today, that organoids have helped us to investigate a mechanism of exit from quiescence and exit from differentiated to a proliferative state, and they allow us to identify, and you know why the epigenetic remodeling is being essential for this transition. Organoids have helped us to identify that cell-cell contacts are important. And this is what indeed determines the behavior of the ductal epithelium. And finally, I've also shown you how using liver organoids to model a particular disease, in particular liver cancer, has helped us to understand a mechanism of how two genes are involved in the disease. And with that, I would like to finalize by thanking all the people present and past members of the lab, which I've been mentioning all along my talk, particularly Anna Dobach and Lucia Cordero and Luigi Aloy and Michael Mackay, as well as Laura Broutier. Of course, our collaborators and our funding. And just to let you know that we are looking for postdocs if someone is interested. So thank you very much for your attention, and I'll be very, very happy to take on any questions.
Thank you.
Thank you, Mary, for your insightful talk, showing us like how liver organoids can be used to understand the mechanism of human liver regeneration and cancer. So I will take the privilege to ask the first question. So I'm curious about the dynamic cell context between mesenchymal and ductal epitheliums during regeneration. And I can see that you show that in the in vivo system, like the ductal cells proliferate first and followed by the mesenchymal cell change. And also in the in vitro, in the organoid system, you see that the cell count direct contact dominant for the growth factor supplement. And just wondering like how this process going, and can you let us know more mechanism about this process?
So we are still investigating on that front. So we think that in vivo, most likely there is a trigger caused by maybe, so this ductal epithelium and the mesenchymal cells themselves, they also contact the neighboring hepatocytes, which we don't recapitulate in vitro yet. And we think that maybe and these are the hepatocytes are the target cells of the toxin that we use to induce liver damage. So we think that maybe the loss of the contact, because this cell dies because of the toxin during the loss of this contact with the hepatocyte, could be one of the triggers that also activate the ductal cells to proliferate, while the mesenchymal cells, for whatever we still don't know, are still in place. And that could be the moment one cell starts to proliferate, then it automatically changes the ratios. This is one of the things, hypotheses we are looking at the moment. We are investigating the cell-cell interactions between the two. And in vitro, since we are missing still this other epithelial cell, it might be that we are not recapitulating. We are not recapitulating the death of someone of a cell in vitro. That could be one. This is one of the ways we are thinking about the problem. And for that, of course, we are trying to make more complex structures at the moment.
Thank you. And I will pick a question from the audience. So this question is there a metabolic plasticity during the differentiation quiescence process?
Oh, very interesting question. We are starting to go in that direction. And yes, I would say it's very, very interesting. Oh, yeah, we think so, yes. We actually was one of our top hits was either metabolic, our top hits when we were looking at the dynamic changes on transcription over the organoid formation or in vivo. We had two top hits. One was related to DNA and chromatin modifications, and the other one was related to metabolism. We went for epigenetics and DNA methylation because it was one of the top hits, but we could have gone on the other direction, and now is where we are going. Indeed, yes, we think that metabolism is very, very important in this transition.
Thank you so much. And I think for time reason, we will move on to our second speaker, and Zhenjie will introduce our second speaker, Dr. Hans Clevers.
Thanks a lot, Chen. So I'll take the honor to introduce our next speaker, Dr. Hans Clever. Dr. Hans Clevers obtained his MD and PhD degree from the University of Utrecht in Netherlands. His postdoc work was done with Dr. Cox Terhoeks at Dana-Farber Harvard University. Dr. Clevers has over 750 publications and over 90,000 citations. He is a member of Royal Netherlands Academy of Arts and Science, and American Academy of Arts and Science, and the National Academy of Science of the United States. Throughout his career, he has worked on the role of Wnt signaling in stem cells and cancer. His major discovery include TCF as the nuclear Wnt effector, the role of Wnt in adult stem cell biology, and of Wnt pathway mutation in clone cancer. LGR5 as a marker of multiple novel types of adult stem cells and as a receptor for the Wnt amplifying R responding. And finally, a method to grow ever-expanding mini organoids from LGR5 stem cells derived from a range of healthy or diseased human tissues. As of March this year, he is now the head of pharma research at Roche in Switzerland. Today, he will give us an exciting talk on organoids to model human disease.
Thanks very much, Zhenjie, for a kind introduction. I'll try to share my screen. I think that works. Yeah, so the previous speaker, Mary Hoek, already gave a good background to organoids, I think. The various types of organoids that have been developed by multiple labs over the past 12 or so years. So what I'll try to do is give you some examples, mostly in the infectious disease space of how various PhD students and postdocs in my lab in Utrecht have been applying organoid technology. So first example I want to give, it was published a few years ago. We did this in our lab, Inya Hio and DJ Dutta mostly, with a collaborator in the US, Rob O'Connor. And what we tried to see if it was possible to use organoids to culture pathogens that were previously not culturable in the lab. And there was one example already published by Mary Estes, the norovirus, very infectious for humans, but never culturable in the lab. It turned out that norovirus requires fully differentiated enterocytes to propagate itself, and they don't occur in cell lines, but they do occur nicely in normal organoids. And as I'll show you in the next few slides, Cryptosporidium does the same thing. So Cryptosporidium is a parasite that causes chronic bowel disease, mostly in third world countries. It can spread from cattle to humans, and it's related to malaria. So it has a complicated life cycle. It's a eukaryotic parasite, but that can go through asexual stages, as you can see here. I won't go into much detail here. And then through a sexual stage will lead to a zygote and then complete its life cycle, which in total would take about 5 days or so. Unlike malaria, there's not a vector, so it goes from essentially from mammals to mammals directly. And to capture this in the experiment in a little movie, here you see a human small intestinal organoid. The small intestine is the site where Cryptosporidium would probably live. So we can inject the oocyst, which is the infectious stage of Cryptosporidium, inject them into the organoids. As you can see, this is really equivalent to the normal lumen of the gut. And it's lined by all of the differentiated cell types of the gut. You see these oocysts land here. Inside oocysts, there are 4 sporozoites. You'll see them soon leaving this oocyst. It's happening here. This is the first stage. So now these sporozoites try to infect a human cell, and it turns out that like a norovirus, they really prefer the enterocytes, so the mature cells on the villi of the gut that normally take up nutrients. So they immediately form the structure that you see here, a meront, and out of this structure, merozoites come out, type 1 merozoites. So it's an amplification step. They enter another amplification step. So you can see that the merozoite type 1 again infects an enterocyte here. Now it forms a type 2 meront, again filled with a number of offspring. And now we enter the sexual stage of the life cycle of this parasite. We're now about 3-4 days into the cycle. In the front, you'll see the male gamete, the microgametes formed. In the back, you see the macrogamete, which would be the female. Microgametes leave the microgamete. They fertilize, as you can see here, the macrogamete, and then presumably they will form a new oocyst. You see that happening here. The oocyst is then released from the epithelial cell, leaves the body of the infected animal or human being, and is then ready to infect the next host. So this is the idea of the test we wanted to do. This worked out very well. Just one slide. We can follow the different stages by confocal, but also by EM that you see here. And essentially by EM, we see all of the different stages as predicted to occur in these organoids, in these infected organoids. And when we wait 5 or 6 days, we can then harvest the oocysts, so the infectious stage on the bottom right. And these oocysts will then, you can see them here, are then ready to infect the next animal. And indeed, from human intestinal organoids that are infected, 6-7 days later, we can actually now infect real mice with the oocysts that have been formed. Just one slide on another project done by a PhD student who's now with Kim Jensen, Katie Romassar. What you see here are cervical organoids from the cervix epithelium. They are quite easily grown from pep brushes. Basically, a pep brush will give you a number of these cervical organoids. They are stratified epithelium, so there's no real lumen. The proliferative cells are on the outside, and the differentiating cells, these keratinocyte-like cells, move to the inside of this pearl-like structure. When we infect the culture with HSV virus, and this is a version that expresses the RFP, red fluorescent protein, and I'll play the movie once more, you can see how incredibly efficient the virus, in a matter of 2 or 3 days, you can see it happening top right, infects and kills these organoids. And we've done this, and many other labs have now used organoids to test infection of other viruses, and I'll give an example of COVID towards the end of my talk. Mary already alluded to the hepatocyte organoids that were set up in the lab in Utrecht by a successor to Mary when she left for Cambridge, Hui Li Hu, a Chinese postdoc who took up the very complicated task to see if she could develop culture conditions to grow from liver not the cholangiocytes or the bile duct cells that Mary just showed, which then turn into bipotent cells, but to directly culture hepatocytes and see if you could capture them in a proliferative state. And this was inspired by the observation by many labs in the past that when a surgeon removes a large part of a liver from a mouse or a human, but leaves the remainder of the liver intact, that then the hepatocytes will rapidly enter the cell cycle, and in a matter of 1 or 2 weeks can rebuild the liver, the part that was gone. And this can be up to 2/3 of a real liver. So this is a massive expansion. You would not call these cells stem cells. They are differentiated hepatocytes, but they proliferate well. And here they do so in the context of human organoid culture. So what you see here are fully mature hepatocytes, so these very large cells. This is a single confocal plane here. You see very large nuclei inside a big nucleus, and we stain here for a serum protein that's abundantly produced by hepatocytes, alpha-1 antitrypsin, and also for a mature hepatocyte transcription factor, HNF4 alpha. So by all means, if you do single-cell sequence, for instance, on these organoids, they look like mature hepatocytes. But I stress that they're not really normal mature hepatocytes because they divide, and here we capture a mitosis. So this was set up by Hui Li and published, I believe, in 2018 in Cell. Really a beautiful paper. Hui Li is now back in China and runs her own lab. Another Chinese postdoc, Daizong Wang, took up this model system. He did a PhD with Ariel Zhang from Shanghai. And Daizong in my lab is involved in a number of projects, but one is to build a model for chronic hepatitis B infections. And as you probably realize, hepatitis B is a major issue in many, many countries around the world, including in China. In some infected patients, the disease becomes chronic. So apparently the immune system cannot get rid of the hepatitis virus. It actually, as an epichromosome, it'll form this what's called the cccDNA. So the virus genome is very stably embedded in the host nucleus. There were no good chronic models for hepatitis B, but here Daizong has managed to grow the organoids of Hui Li actually in 2D, so he expands them in 3D, he plates them out in 2D so that they no longer proliferate. Now they're mature hepatocytes, but he can keep them alive. You see here a month, but we can now do it for several months. And when he infects these 2D cultures of hepatocyte organoids, you can see bright expression, in this case of the hepatitis C, hepatitis B core antigen. There is secretion of the HbF. And we can readily demonstrate by PCR that the genome of these viruses persists in this form of the cccDNA, but we even see integration of the viral parts of the viral genome into the human host cell. So we hope to use this, just one slide, but to hope to use this as a platform to now study the chronic state of infection and possibly contribute to the development of drugs for this particular, no, this movie doesn't play. Hold on, for this particular devastating disease. So this is a paper that will come out in the next few months in Nature Biotech, mostly done in Andriel's lab. You see your name here. First author is Florijn Dekkers. My lab was involved, and Jürgen Kubel's lab in Utrecht was also involved in doing this. And what you're looking at are, in yellow, are human breast cancer organoids. The paper has a number of different lines from different patients, and they are confronted with, in white blue, with human T cells, peripheral blood T cells that have been transduced with a V gamma, V delta T cell receptor, of which it was known, and particularly from the work of Jürgen Kubal, that this receptor in some way distinguishes between normal cells and tumor cells. So if you put this receptor into normal T cells, CD4, CD8 T cells, you make them cancer specific, and they can see a wide range of different cancers. Here they're seeing breast cancers. The yellow organoids are the cancer organoids when they're viable. When they're killed, they turn red, as you can see here. And what Anne does, she creates these movies that run over 1 or 2 days. Very high volume movies, large amounts of data, but this allows her to actually go into these movies and then classify the behavior of different T cells in their effect on the breast cancer. So some T cells don't move at all, some T cells move around, but they never encounter a breast cancer organoid, but some actually will start killing. And now when you look at these organoids, at these T cells, you can classify them into different behaviors, and I'll show you in the next 3 movies, I'll show you the different behaviors. So here's sort of an average T cell that manages to attach to an anchor point to the yellow breast cancer organoid. And you might appreciate that it sticks out these long extensions. And in a matter of less than 24 hours, bottom right, you see the clock running, it actually destroys the entire organoid of maybe 20 to 25 cancer cells. And it's just a single T cell that kills sequentially 25 cancer cells. I don't think this has ever been imaged. And this is really the strength of Andreas's lab that she can visualize this. Here you see another one. So T cell comes in from the left, and you can see actually it creates these long extensions. And so it stays on one side, but at a distance, using these slings, these lassos, it can actually slowly destroy this organoid. Now, the movie unfortunately stops, but I'll go to the next movie. You see that here. And this is the most active T cell. Ant calls it the serial killer cell. You see it drills through these organoids, kills a number of cells, runs away, runs to another cancer organoid, kills a few more cells in this organoid. It's now only 5 hours in of the experiment. It runs to a third organoid, again rapidly kills 3, 4, 5 cells, and then disappears into the sunset. Now, these T cells are extremely interesting for clinical use, if you would want to use them as CAR T cells. And Ant has been able to actually separate them from all the other T cells by, you can see it actually drills into the organoid. If you now spin down the culture, the organoids, you have trapped these most active serial killer T cells inside an organoid, and that's when you can do single cell sequencing on the T cells. And in the paper, you might want to look it up when it appears, we now have a very nice signature, and has a very nice signature for this particular type of T cells. And hopefully with this signature in hand, it'll become possible to, when one creates CAR T cells, to increase the numbers of T cells in this prep of CAR T cells that have more of this signature, because they are probably the ones that are best at killing tumor cells when introduced into patients. Two more stories. One was published recently. It was essentially work from 3 PhD students who also inspired this talk, Cayetano and Jens in my lab, and Axel in Ruben van Boxel's lab. And these 3 students have noted that there are different strains of E. coli in the human microbiome, and one strain that is quite common, probably 20% of all people, at least in the Western world, carries this particular version of E. coli in their microbiome, and it's defined by an additional 60 kilobases of DNA. You see that here. It's called the pathogenicity island. There's about 10 genes in this extra piece of DNA, and together these genes constitute a synthesis pathway for a small substance called colibactin. I'll show it in the next slide. So bacteria, E. coli, that have this extra bit of DNA can make a small molecule called colibactin. And when they can make that small molecule, and you culture these E. coli on top of human cells, you'll see that these E. coli are genotoxic. They actually will cause double-strand breaks in the genomes of the human cells. And I'll get back to that a little bit later. So the question that our 3 PhD students asked is the following: is this PKS E. coli strain, carried by about 20% of people in the Western world, is it not only genotoxic, so causing double-strand breaks in the DNA of human cells, probably leading to the death of those cells, but is it, maybe even worse, can it be carcinogenic? So can somehow PKS E. coli induce mutations in the DNA of human intestinal cells? This is this proposed structure of colibactin, very labile, very difficult to work with. You cannot buy it. This beautiful paper in Science describes the structure and also predicts that there are 2 warheads. And with these 2 warheads, colibactin can bind covalently to a DNA helix. And if it would do this on the 2 different strands of the DNA, so covalently bind to an A here, and then covalently bind to another A on the other strand, this is now an interstrand crosslink. This is not compatible with life of that human cell, and presumably in trying to resolve this, the human cell creates a double-strand break, and that's why colibactin, when bacteria can produce colibactin, they are called genotoxic. So here you see the first experiment. So on the left, you see a very potent PKS E. coli strain that we obtained from Michel Bonnet in France. So the left one has the full pathogenicity island, the PKS island. The one on the right lacks one of the genes in this PKS island. It is our negative control. So on the left, you have the bad guy, and on the right, you have the control. Now you can grow these E. coli quite easily on agar plates. You see that here. You can pick them up in needles. And what Jens and Cayetano did is they then injected these bacteria into normal human intestinal organoids, let them in for a day or so. So during that time, they can now secrete colibactin on the left. Of course, the one on the right cannot secrete colibactin because it lacks one of the key enzymes. So that's the control. And then we could simply ask, can we repeat what is seen in the literature, namely that exposure of epithelial cells to colibactin, to these bacteria, leads to double-strand breaks. And that was usually done on cell lines, but here we do it in organoids. So you see about 50 organoids here. Each of them were injected with PKS E. coli. And you can indeed see that inside this particular organoid, there's many double-strand breaks. Every dot is a double-strand break, and we visualize them by staining for gamma H2AX foci in red. The control E. coli, we injected them into all of these organoids, but there was almost no double-strand break. I think we saw a single one in this entire organoid culture. So indeed, we can recapitulate that PKS E. coli causes double-strand breaks. Then the question comes, do they actually, when we allow chronic exposure to these bacteria, do they induce mutations, and are they maybe carcinogenic? So again, we inject, or I should say Jens and Cayetano inject, but now we want to expose over 3 months. So what Cayetano and Jens do, on the Monday they inject, on a Friday they treat with antibiotics to basically get rid of the bacteria. They then can passage the organoids that are growing, and they can do this, go through these cycles for a significant period of time. We now hope to see many mutations here, but not in the organoids that were injected with the control E. coli. However, if we would just sequence these organoids, we would probably not see the mutations, because every cell in this organoid will have a different set of mutations. So what they then did is they subcloned single cells from these organoids, expanded them briefly to get enough DNA for a good quality whole genome sequence. And now, in essence, every subclonal organoid gives us the genome of the cell that was exposed to colibactin. So on the left, we have the offspring of single cells that had been exposed for 3 months to colibactin. On the right, we have our controls. So essentially, we read out the mutations in the DNA, we classify them according to a method that was described by Mike Stratton. And indeed, we see that there are many more mutations in these organoids versus the control organoids, and particularly T2C mutations in a particular background. And if I now know that, take you into a more deep analysis of what kinds of mutations are induced, they're very unique. So it's not just random mutations we see when we expose to PKS E. coli, but we see that the mutations always occur in a sequence context. What they do is they either change a TA base pair that you see here into any other base pair, so that would lead to a mismatch mutation if it would be in a reading frame, or we see that this TA pair is entirely lost, and that would be a single or maybe a 2 base pair deletion, and that would lead to a reading frame change if that would happen in a genome. And it always happens in this particular sequence context. Now, if we now go to the model of how colibactin interacts with DNA, what we think is happening is that one warhead of colibactin will bind covalently with this A, so that would be this A here. The other warhead here binds to the A that's opposite this T, so the A on the other strand. Now there is a double-strand crosslink. The cell cannot live with this crosslink, has to get rid of it. It probably produces a double-strand break, or maybe in another way, what it does, it gets rid of colibactin, but in that process, it either changes its base pair into any other base pair or deletes this base pair. And both would be a real mutation that would then be given on to the next generation of cells. Now, this is very artificial, but it was a very unique signature that we found there. We call it the PKS signature. So an A at -3, a TA at base pair at position 0, and a TA base pair is either gone or it's mutated into another base pair. And we then asked in a very large collection of of cancers of a large number of different origins. As you can see here, we asked which of these cancers show evidence of this particular type of mutations. And lo and behold, we essentially only see these mutations in colon cancer. It's about maybe 10%. We have better algorithms now. We probably find them in up to 15% of colon cancer patients in large cohorts. We find that many of the mutations in these cancers appear to be caused by colibactin because they have this particular A at -3, TA at 0 that's changed or deleted. We get the occasional tumor that has a signature that's not colon, but it's head and neck or bladder. We have a few more, but these are also sites where we know that bacteria, particularly E. coli, can live. And these observations were confirmed then in a large cohort from Genomics England, more than 2,000 colon cancers. Again, about maybe 10% or so of the cancers show evidence of PKS E. coli activity in the cancers. And then we asked, do we see these mutations not just randomly in the genome, but do we see them in cancer genes? And particularly APC and also P53 are very good targets for this mechanism. And just to convince you of that, here we can show you a large number of mutations in these colon cancer cohorts that specifically seem to hit the APC tumor suppressor gene. And I should probably explain to you that APC is a tumor suppressor that's mutated in up to 90% of all colon cancer patients. So the most common mutation in colon cancer. It's also presumably the first mutation. So if you knock out APC in a normal stem cell in colon, it'll make an adenoma, and very soon afterwards, it actually will become an adenocarcinoma, and then it's really malignant. And apparently, the APC gene can quite easily be disabled through this particular process. So to summarize briefly, PKS E. coli induces mutations. It's not only genotoxic, it can cause single base substitutions. It also can cause small deletions. Both are seen in colon cancers in a subset. And this has implications maybe that once you try to find out who carries these bacteria, and particularly if you are an individual at high risk for colon cancer, it's probably not a good idea to let these bacteria persist in your microbiome. And they can be removed quite easily with a simple antibiotic treatment and possibly with a fecal transplant. And then finally, I need another 5 minutes or so. When COVID broke out in Holland, our institute went into quarantine. Went into lockdown in March of 2020. We were allowed to do COVID experiments. And one idea that came up was the following. We realized that the receptor for SARS-CoV-2, which we then already known, receptor is also for the original SARS virus, ACE2, is typically expressed at the luminal sites of epithelia in the lung and the gut, but the most powerful, the strongest expression of ACE2 essentially is present on the gut microvilli, on the villi that you see here. That's one. Second, we learned that quite a few patients that are infected with SARS-CoV-2 show GI symptoms, gastrointestinal symptoms, so nausea, diarrhea, stomach aches, and also the first PCR assays became available in March of 2020, and there was some case reports that viral RNA was detectable in stool. So we asked, is SARS-CoV-2 not only a virus that infects lungs, but can it also infect other organs like the gut? And we could do so because, of course, we had all of these different organoids in our lab. And we did this in close collaboration with the lab of Bart Lamers in Rotterdam in Holland, of Bart Hagemans and his PhD student Bart Lamers. But you can see here that indeed the virus readily infects human intestinal organoids. It also infects airway organoids and alveolar organoids from lung, but we get the best propagation of the virus in intestinal organoids, a log4 increase of live virus in less than 3 days, and also in viral RNA titers. When we stain, we actually, by EM, I don't show this here, but ACE2 would be on the luminal side of the organoids, like it would be on the luminal side of the gut. So the virus comes in through the lumen of the lung or the gut. It then, because ACE2 is there, it can enter the cell. We stain here for the nuclear protein of the virus in white, so a single cell is infected. By EM, we see that the viruses are extruded again through the apical site into the lumens. And indeed, when we wait 2 days or so, we now see that almost every cell in an infected organoid has undergone an infection. But when we did not infect an organoid in the first day, you can see there's no viral infection because the virus doesn't go out at the basal site and cannot enter through the basal site. It always goes through the luminal site. And I stress this because for the final few slides, it's going to be very important to realize that these viruses always enter through the apical sites of fully polarized epithelial cells. We did a lot of single cell sequencing, and we found what many other people have published, that there's defects in interferon responses, etc., etc. That was not so interesting, but we then went on, this paper is now actually published, to study what kinds of host genes are needed for viral propagation. And we could address that quite easily because particularly in gut organoids with CRISPR, one can knock out and make mutant clones, subclonal organoids. Sequence confirmed that there really is no expression of wild type protein anymore. And we've made hundreds of these, we knocked out hundreds of these genes or knocked in intestinal organoids, meanwhile, in the lab. Here, very simply, we knocked out ACE2. There's 2 clones in blue. And indeed, when we knock out the receptor, the wild type cells are here. The virus can no longer infect cells that are mutant for ACE2, as expected. The original SARS virus, same thing. Here see wild type intestinal tract organoid. We knock out ACE2, the same 2 clones. There is no infection. MERS, a third member of this particular kind of coronaviruses, is known to use another receptor. And when we knock out ACE2, it happily infects, as you can see here. Vice versa, MERS is known to use DPP4. When we knock out DPP4, MERS virus no longer infects, as you can see here. DPP4 has also been proposed to be a receptor for SARS-CoV-2, but that's not, at least it's not essential, because when we knock it out, SARS-CoV-2 here happily infects in a matter of 2 days. So this all confirms what was known. We then went through a long list of proposed human host genes that the virus uses, and these mostly came from several papers doing genome-wide CRISPR screens. Typically, these were done in the workhorse cell lines of virology labs, like Vero E6 cells. They are not your polarized epithelial cell, they're more like fibroblasts, and Vero E6 cells are African green monkey kidney cells, and when you look at them, look more like fibroblasts. And much to our surprise, we already confirmed ACE2 as a key human gene. We confirmed TMPRSS2, that you can see here. And TMPRSS2 is a surface protease, I'll show it in the last slide, that activates the spike protein. But we could not confirm any of the other, and there's a number here, but there are many more in the paper. We could not confirm any of the other genes. And for instance, one in particular, cathepsin L, a big hit in these genome-wide CRISPR screens. If we knock it out, and these are clonally confirmed, 2 different clones, we even get more infection than we would get with the wild type that you see here. We also observed that chloroquine, which, as you all realize, has been proposed as a great drug for COVID in 2020, and hundreds of thousands of people were given chloroquine with no effect, no beneficial effect, but with side effects. So obviously, chloroquine in our hands is a very good blocker of infection. You can see that here. IC50 is at 1 micromolar. Total block of infection is at 10 micromolar. And we take this concentration that we now added to our organoids, and we try to infect. Infection is happily continuing. There's no effect of chloroquine. So to summarize this, and then I'll finish. The virus, we believe, in these non-epithelial cell line-based systems will bind to ACE2. ACE2, by the way, often have to be transfected into those cell lines because they not naturally express it. It's really an apical surface marker of epithelial cells. But when you put it in, the virus will bind. It's then endocytosed. Endocytosis is sensitive to chloroquine, and that's why this drug probably works so well in these Vero E6 and similar models. It ends up in an endosome, and it then is released through the action of cathepsins. I just showed you that cathepsin came out of the genome-wide CRISPR screens, and so this would allow the action of cathepsin L or B, allow the extrusion of the viral genome into the cytoplasm of the host of the human cell. This happens in non-epithelial cells. Epithelial cells naturally have ACE2 on their surface. So in organoids, you have ACE2 on the right side where the virus normally enters the human body, which is to the apical surface. The virus can bind like it would do here, but now TMPRSS2 is also present. So this is the surface protease. It'll cleave the spike protein, activate the spike protein. Now the virus envelope directly fuses, it merges with the host cell membrane, and now the viral genome is directly injected into the host cell. So there is no endocytosis needed for this particular step. And that's why hydroxychloroquine probably is fully ineffective in natural infections by this virus. So I think a few take-home messages. First is I think that it's always interesting to not only use cell lines, but also use organoids if you want to study diseases, particular infectious diseases, particularly the intestinal infections or the airway infections, because there the viruses by definition have to come in through the lumen, and the only part of the human they will encounter is the surface epithelium, the apical side of the surface epithelium. And second, I think it explains why this drug hasn't worked well, if at all, for COVID patients. And with that, I've reached the end of my talk, and I'm happy to stop share and give the word back to Zenji.
Thank you so much, Dr. Cleavers. It's a very inspiring talk. So I will take the privilege to ask the first question. So for the PKS positive E. coli story, so I wonder if you have determined which receptors from the intestine organoid system that would be responsible for the interaction.
Yeah, so that's a key question, because we believe that colibactin really has to be inserted, has to be injected into the host cell. It's very labile; it probably will not travel. So we think that PKS E. coli has to colonize crypts to be as close as possible to the stem cells, and then either inject colibactin, they have injection machinery into the stem cell, or maybe the entire bacterium invades. That would be another mechanism we're currently trying to establish, invades the stem cells. And what I didn't say, but when we published our paper, where we in a very artificial way found this unique mutational signature, this is a Nature paper, but there was also a paper by Mike Stratton, who basically didn't look at bacteria, but he sequenced crypts of individuals. And what he would find that in some individuals, he finds the exact same mutational signature in the crypts, in the stem cells of human beings, but he would find them in 2 or 3 adjacent crypts, and then in some other crypts, if those mutations would not be present. And so his prediction is that there is some mutational effect that's very local, and that comes from the outside, which can happen in one crypt, but not happen in the next crypt of a human being. And so that would fit very well with our bacteria, because we think now that the crypts that Mike Stratton saw that had the mutations probably were invaded by colibactin, by E. coli, by this bad E. coli. And the neighboring crypts that didn't have the signatures were not invaded by this PKS E. coli. It's currently under investigation, but yeah, I think this is a very good question.
Okay, so another interesting part is for the intestine organoids. So people would think of the microbiota, which in recent years has been demoed would greatly affect drug pharmacokinetics or therapeutical effects. So I wonder, you talk about those virus, E. coli, or that would cause the DNA damage. So any evidence those microbiota actually instead will protect from intestinal organ from the DNA damage or those defects?
Again, a very good question, but of course the microbiome is a very complex mix of many bacteria and strains of bacteria that we even usually don't look at. So our approach has been to use single species and mutants of that species. But we are actually in a CRUK Grand Challenge grant where other labs are working with communities. So maybe 10, 12 different species of bacteria that they can stably propagate in the lab. And we have been collaborating with these labs to see if you now take the secretions, take the supernatant of those microbiota that are closer to the real microbiome, do we see effects if we culture organoids in those complex mixes? And yeah, we see all sorts of effects, but it's going to be not simple to basically, find cause and effect there. But yeah, I think by we have shown you can do a single bacterium in an organoid and make a small discovery. I hope that other people will start doing this also with more complex mixes of the microbiome, and then address types of questions you're addressing.
So we got a few questions from the audience, but probably due to the time issue, I'll just pick one that is a brainstorm type of question. So whether it's possible to use a organoid in longevity study.
Yeah, you can actually grow organoids for a long period of time. You can grow them for many years, but I don't think you want to do a PhD on growing organoids for 90 years or so. But you can actually grow organoids from people of very different ages. And that has been done. And actually what you see, for instance, is that there's a linear increase of mutations, maybe as expected. And we know for gut, you get about 20 mutations, single base changes per year of life. Or sorry, I should say 40, so about 1 per week in a stem cell. And if you're 80, you can calculate how many mutations you have in every stem cell in your gut. And that's probably mutations changing the DNA are probably a large part of aging. And yeah, so and species actually that live long or live short will have very low levels of mutations or very high levels of mutations, as recently published. So these types of things you can address in organoids. Yeah.
Okay. Once again, thank you so much for Dr. Cleaver's exciting talk. So we, due to a time issue, so we have to move to the third speaker, Emma Rollins. So Chen would do the introduction. Thanks again.
Thanks, Junjie. So it's my great pleasure to introduce our next speaker, Dr. Emma Rollins. Dr. Rollins obtained her PhD in developmental biology from the University of Edinburgh in 2002. Then she performed postdoc work with Professor Brigid Hogan at Duke University, where she identified stem cell populations in mouse lungs. In 2009, she started her own lab at the Gordon Institute, University of Cambridge, and in 2020, she was promoted to senior group leader. Her lab works on lung stem and progenitor cell biology, combining innovative human organoid models with mouse genetics. Among the years, Dr. Rollins has received several awards, including the Sherry Tickle Medal from British Society of Developmental Biology for Female Scientists, and MRC Centenary Award and Career Development Fellowship. Her talk today is Building the Human Embryonic Lung, Lessons from Organoids. Let's welcome Dr. Rollins.
Thank you very much, and thank you very much for inviting me to speak today. I'll also try and share my screen. I hope this is working. Here we go. Is that looking okay? Great. So thank you again. And I'm going to tell you about our work using human organoids to study human-specific aspects of lung development. So why be interested in lung development itself? Well, we're interested in improving in vitro systems for disease modeling and drug discovery. We're very interested in lung regeneration and the question about whether developmental mechanisms are reactivated in lung cancers. So how does the lung form? I'm sure you all know that lung formation occurs by branching morphogenesis. But the key thing you need to know for this talk is that the distal epithelial tip cells of the branching lung are a multipotent progenitor, initially generating the airway epithelium and at later stages generating the alveolar epithelium. So what does that look like in a human? So this is a section of a 7-week human embryonic lung. We're very fortunate to be able to obtain human embryonic and fetal material from a tissue bank based in the UK, the Human Developmental Biology Resource. So when you cut sections to these lungs, you can see the tips at the edge of the lung here. The tips are here, SOX9 positive in green. So they're undergoing undergoing self-renewal and growing out in this direction. In fact, you can see this tip is branching. And as cells get left behind in the stalk region, they start to differentiate. And even at 7 weeks, these cells are already requiring different cell fates and are surrounded by a layer of differentiating smooth muscle. So a number of years ago, I had a wonderful PhD student, Marco Nikolic, who now has his own lab at UCL. And we wanted to ask the question, how similar are mouse and human lung development? And Marco did an awful lot of characterization. And really, the key things are boiled down here. We all know that mice are not just fairy humans. Obviously, the size is very different. The cell composition and organization along the airway tree is quite different. But actually, what's really key to me is that the molecular mechanisms have different details. And if we're interested in using our work in the future for drug discovery, then it's these molecular details that are going to be the key. So because of this, we're inspired by the work of Hans and Mary, and actually had help from Mary's lab while she was at the Gurdon Institute to set up our own organoid system, which is shown here. So we take our favorite undifferentiated progenitor cells from the human epithelial lung. We plate them in Matrigel, provide the growth factors that they need, which you can find in the paper, but primarily FGF and Wnt need to be activated. And if we do that, we can self-renew and expand these cells. So they massively divide, they maintain their undifferentiated fate, and you can see actually at the end of the first passage, we have lots of little buds that look remarkably like the starting material. So this is great for us, and I think the way of thinking about it is analogous to an embryonic stem cell line, expanding and self-renewing your stem cells, but now this is unique to the human embryonic lung. But of course, we're not interested in the stem cells themselves, we're interested in how to differentiate these cells, and we can do that very nicely to airway epithelial cells fate or to alveolar fate. This is our latest airway epithelial protocol. You can see it's very beautiful. This is our alveolar epithelial protocol from 2017. What I'm going to show you is how, later in the talk, is how we do it now and how really these cells properly form. So how do we study then human organoids in order to study human lung development? Well, we still use the mouse, and the mouse is always going to be key to a lot of our research. But in terms of the organoids, what we like to do is flip-flop backwards and forwards between describing the human embryonic tissues in detail and performing functional experiments in the organoids. So one type of description we've been doing recently with collaborators is what everyone else is doing, the big single cell sequencing experiment. So we have a single cell RNA sequencing for various different cell types within the lung, in fact, we think all cell types, combined with spatial chromatin accessibility, spatial transcriptomics, and very detailed in situ protocols. And of course, combining this with the organoid work to test specific hypotheses. So I mentioned this briefly now, not to go into the details. We have some very interesting things in here. But what you're going to see is this work is going to come back throughout the talk and how we're using it to do the apply it to our functional questions. So the two things I really want to talk to you about today are what are the transcription factor networks that coordinates tip cell self-renewal of our organoids. And this is going to allow me to highlight the work of a PhD student, Darway's son. He's now moved on to a postdoc at the Broad Institute in Boston, and allow me to highlight the genetic modification tools that he developed for our organoid system. And secondly, the question of how do human lungs coordinate alveolar development, which can allow me to highlight the work of our postdoc, Kung Tae Lim. So what's the transcription factor network that coordinates tip cell self-renewal? So in order to answer this question, Darway had to develop a lot of genetic tools that work very efficiently in our organoids for knocking down, overexpressing, or making knock-ins to manipulate the intrinsic cell behavior of the cells rather than manipulating the external medium. And I'm going to talk to you, we published this in last year, and I'm going to talk to you about particularly the inducible knockdowns, CRISPR-I, because we're using this, I think, in virtually every project in the lab now. So I'm sure you all know the CRISPR-I system. So this is dead Cas9 molecule, which is fused to a Crab domain and can spread heterochromatin across the region of DNA that the guide RNA targets it to. So what we're doing is introducing it to the organoids using lentiviruses. One lentivirus containing the CRISPR-I system under the TET3G doxycycline-inducible promoter, and one containing a constitutive guide RNA. And in both cases, we're labeling with a fluorescent vector for efficient selection. But the key thing here, Darway's key innovation, is to fuse the whole system to this DHFR protein destabilization domain, which now gives us extremely exquisite control of the timing of knockdowns. So in the presence of the two drugs, we have a very tight knockdown here, for example, of CD71, the surface molecule, and this works out for many different genes at the genome, though it's true to say not all. So with this in hand, we can then go on and ask Darway's key question, which is which transcription factors are required for progenitor cell self-renewal? And we chose to perform a pooled CRISPR-I dropout screen for this self-renewal phenotype. And in particular, we wanted to know how well would this work in the organoids. And this is a proof of principle assay for us, because obviously we want to go on in the future and apply this to various different lung disease phenotypes. So in this experiment, we're taking established organoids, we're single celling them, and now transducing with two lentiviruses, one containing the CRISPR-I vector and a second containing a single guide RNA. So we deliberately made a relatively small library for this proof of principle test. We want to know how well this would work. And we chose 49 transcription factors, 4 positive controls, 5 guide RNAs per each gene, and I think 33 negative controls. So you transduce the cells and select the doubly transduced cells. We expand those into small organoids, turn on CRISPR-I, and passage them. And then, of course, the question is, if you sequence the guide RNAs at the beginning and the end of the experiment, has guide RNA abundance changed? Do their guides increase? Do they decrease? And what does this tell us about gene regulation? So the key point I want to make here is this was tremendously reproducible. So we did both technical and biological replicates. Our technical replicates, I'm happy to say, are as good as a cancer cell line, really the R-squared value of almost 90%. Whereas our biological replicates, as you'd expect for primary cells, are a lot more variable. And I really start to think about this in 2 ways. How do we take advantage of this biological variability to explore further human genetics? But in this case, we're taking advantage of it to pick things that are particularly well conserved between our different organoid lines. So I don't have time to show you the full details of the screen, and I'm just going to tell you that SOX9 emerged as a very strong dropout here. So very few organoids at the end were mutant or depleted for SOX9 protein. So is SOX9 actually required then for human lung progenitor self-renewal, which is what's predicted by Darway's screen? So now we can knock down SOX9 individually. You can see it's knocked down very effectively by the guide RNA system, and in 6 different organoid lines, we can no longer passage them over time in a serial passaging assay. So really suggesting that, yes, it is a self-renewal key factor for organoid self-renewal in these lung epithelial embryonic organoids. So is it truly a self-renewal phenotype? So to address that, Darway did a bulk RNA-seq experiment. So just highlighting some key genes here. So in our control, we have very high rates of cell cycle, very fast cycling cells, and these cell cycle specific genes are lost in the SOX9 knockouts or SOX9 depleted organoids. Whereas actually a whole set of genes that actually in our single cell RNA-seq data are expressed in differentiating secretory cells, which the first cells differentiate in the lung begin to be expressed in the organoids. So really here we've got 2 effects. The cells stop proliferating and they initiate premature differentiation. So strongly suggesting this is a true self-renewal phenotype. So this is great, but SOX9 is a transcription factor. So the key question is, what's it actually directly binding to and directly affecting? So our idea was to do this and answer that question using ChIP-seq. The issue we ran into was that the antibodies we could obtain for SOX9 were just not of high quality enough to be for us to be confident with our data. So what we turned to instead was a collaboration with my good colleague Andrea Brand to use her targeted DAM-ID system in the organoids. So Andrea has really pioneered this very extensively in fruit flies, but now applied it to many, many, many other systems. Actually, Mary has also used it. So we applied it to the human embryonic lung organoids. So we're again introducing the vectors using a lentivirus. Then you express a very low level of the fusion protein of interest between your favorite gene, in our case SOX9, and the DNA adenine methylase from E. coli, this DAM protein. So the idea is that SOX9 is going to bind to its normal binding sites. It's going to hemimethylate the nearest GATC regions. We can cut those with a restriction enzyme that's sensitive to hemimethylation and generate DNA fragment library just containing SOX9 binding sites. So of course, we can overlap with our RNA-seq data to produce a list of genes that are only bound by SOX9, but whose transcription changes when SOX9 is not bound. So this was very exciting. We have our list, but this also gives us an opportunity to test how well the organoids are actually modeling what happens in vivo, because we have the whole transcriptome of the tip epithelial cells from our single cell RNA-seq library. So you can make a very, very strong prediction here that SOX9 is only expressed in the tip cells in vivo. So any genes that are directly activated by SOX9 should be expressed in human tip epithelial cells in vivo. So we can test that in 2 ways. One, by going back to the single cell data and projecting the genes from the RNA-seq and the DAM-ID experiment onto the single cell data, and indeed they're expressed in the tip epithelial compartment. And secondly, we can do a more conventional approach and go in and just check each gene expression using RNA-seq or CD44. CD412, which is or protein. So I don't have time to go into all of the details of what SOX9 is binding to and activating, but actually two key things came out that we were very excited in. And that these are FGF and Wnt activators. So you can see them illustrated on the slide here. ETB5 and ETB4, which are well-known FGF downstream proteins, are transcriptionally activated by SOX9. And LGR5 is also transcriptionally activated by SOX9 in the TIP cells, again, the Wnt agonist. So just to very briefly show the model, we now think that SOX9 activity is actually locking the cells into the self-renewing progenitor state. So Wnt itself directly activates SOX9 transcription, but SOX9 feeds forward into this by amplifying the self-renewing signal and activating LGR5 transcription. SOX9 also activates EDV4 and 5, again activating this or amplifying this FGF receptor pathway, which again is also crucial for self-renewal of our tip epithelial cells. Together, SOX9, ETB4 and 5 activate proliferation set of genes. I'm not showing the ETB4 and 5 DAM-ID data, whereas SOX9 itself is actually much more important here, and it also represses the differentiation, particularly of the first secretory cell genes to appear in the lung. So SOX9 is really locking these cells into the self-renewing progenitor program. And I think you can start to ask yourself, how do they ever leave? And I'm happy to chat about that later if anyone's interested. But this, I think, is a really nice example of how we can now use this CRISPR-Rise screening to identify, to pick apart very many different pathways using organoid type systems. So we move on to answer my second question, which is how do human lungs coordinate alveolar development, which is the work of my postdoc, Kung-Tae Lin. So everything I've been telling you about so far has been from the airway branching stage of lung development. So lungs start to form in the human about 5 post-conception weeks as little buds from the foregut endoderm. They branch and branch and branch to make the airway tree. And all of our published organoids so far, and what I've been talking to you about, are derived from this branching stage of lung morphogenesis, and they differentiate beautifully into airway epithelium. We can get them to differentiate into alveolar epithelium, but they really don't want to. You've really got to push them very hard. But interesting, the lung starts to build its alveolus from about 16 weeks post-conception, and we can obtain samples up to about 22 weeks. So at this period, the tip epithelial cells look quite different, and this is the little region here is the first region of alveolar differentiation in the developing lung epithelium. So we hypothesized the tip epithelial cells would be different at the airway branching versus the alveolar formation stage. And of course, we can check that using our single-cell RNA-seq data. This is just a UMAP of the epithelial cells, and you can see that the tip cells actually move with age across the UMAP. So we separate them here into early, mid, and late stages. I like to point out that the late stage tip cells are very closely connected to the alveolar type 2 and the type 1 epithelial cells. John Marioni would be very cross if I pointed at UMAP and said that. And he would much rather I show you a force-directed embedding plot of this data, where we can see that the mid and early tip cells are connected very nicely to the differentiating airway epithelium, whereas our late tip cells connect very nicely to the alveolar type 2 and the type 1 cells. So you can predict that these mid and early tip cells are generating alveolar epithelium, whereas these late tip cells, generating airway epithelium, whereas these late tip cells are generating alveolar epithelium. But is that the case? So in order, of course, we can test that using organoid models. So let's now plate the late tip cells in the organoid system and ask what happens. So we were very excited the first time we did this, because these are the first organoids we've got that actually self-organize and make multiple cell fates within the culture. So here our tip cells sit at the edge of the dish, and actually in the center, they start to differentiate into immature airway progenitors, when we grow in these self-organizing, self-renewing conditions in the presence of both FGF and Wnt. So we can passage these organoids for many, many passages, and actually it's the tip cells, these tip progenitor cells, that are the ones that are always responsible for making the new organoids. But what about if we change the conditions? Well, if we grow in high Wnt, we get very beautiful alveolar type 2 epithelial differentiation. And if we grow in high FGF, we get very nice airway differentiation. So really suggesting that our tip epithelial cells at this stage are multipotent, but they can very nicely make the alveolar cells, which the earlier stage tip cells really cannot effectively. So we've got this self-organized organoid, which we can passage over time, and we know it has the capacity to generate alveolar type 2 cells. But all these experiments were done on freshly isolated cells direct from the lung tissue. So we wanted to know, if we passage these cells over many generations, could we find a combination of growth factors in which we could still generate alveolar type 2-like organoids? So why is this so important? This is really key because you can grow alveolar organoids from our lungs, from adult lungs, but they're really ineffective. The cells don't want to divide. The job of an alveolar type 2 cell is to sit and spit out an awful lot of surfactant and stop your lungs from collapsing. And they don't want to be infected with any sort of virus. It's a barrier epithelium. And of course, its job is to essentially senesce or die in response to viral infection. Although, of course, we all know that doesn't happen. In many cases, we do get viral infection in the lungs. But in the dish, it's very difficult. So we wanted an expandable organoid system, which we could switch the growth factor conditions and generate alveolar type 2-like cells that would be amenable to genetic manipulation. So Kung-Tae spent a lot of time optimizing these conditions. And the presence of this very specific growth factor cocktail, we can now get actually mature alveolar type 2 cells that secrete surfactant into the lumen of the organoid, undergo a specific cell shape change. And actually on the EM, you can see them generating these beautiful metabodies full of surfactant that they secrete into the organoid lumen. So this is now a very exciting tool for disease modeling. And I'll show you a little bit of that at the end if I have time, but we're using this to explore various different aspects of lung disease, including fibrosis models now. But what else can we use this system for? And what can we use it for in terms of understanding human development and the genetics controlling human development? So something we'd noticed from our single-cell RNA-seq data is that Wnt2 is expressed everywhere in the lung, really everywhere. All these fibroblasts are Wnt2 positive. For those of you who are interested, these are future alveolar fibroblasts. They're adjacent to our tip epithelium, which express high levels of surfactant protein C, which is also a Wnt target. And these cells are also expressing high levels of various other Wnt targets, axin-2 and so on. But as soon as you move slightly away from the tip, the descendants of these cells, they turn off SBC very rapidly. And they also have much lower levels of Wnt signaling. So we were asking ourselves the question, if Wnt is everywhere, what protects these cells from Wnt signal? How do they turn off these genes so quickly? So Kung-Tae noticed in our single cell data that a population of myofibroblasts, which is adjacent to what we call the stalk epithelium, expresses high levels of the secreted Wnt antagonist notum. So in order to explore how this system worked, we started performing a series of co-cultures, either with the tip epithelial cells, the alveolar fibroblasts, or the myofibroblasts. So analogous to what Mary was showing earlier, in this case, we're using just a very simple co-culture system to grow different combinations of cells, either on the basal layer or in combinations. I'm not going to show you the full data set here, but really what I'm going to say is that this really supports the hypothesis that it's the myofibroblasts are sufficient to stop these cells responding to Wnt, sufficient to turn off SBC. And it's very interesting that the differentiating alveolar type 2 cells, as one here, are always located at gaps in the overlaying myofibroblast layer. So strongly suggesting this mechanism is conserved during type 2 differentiation in vivo. So the second thing we've been doing this organoid system a lot is, again, going back to our single cell RNA sequencing data and asking, can we identify transcription factor combinations that are sufficient to promote specific cell fates? So in this case, taking highly undifferentiated organoids from 6-week-old lungs, growing them in the presence of single or combinations of transcription factors and asking what cell fate we get. So it's just one example here. So we turn on the factor NKX2.1, which is a homeodomain transcription factor. You can see we introduce this dramatic cell shape shift in the organoids, and they start to express high levels of our favorite type 2 cell markers, surfactant protein C. Indeed, they become type 2 epithelial cells. So we can take advantage of this system and to do the final experimental data I'm going to show you to ask the question, how do neonatal lethal point mutations in NKX2.1 actually affect development? So these are tremendously distressing conditions. There are heterozygous point mutations in the homeobox of NKX2.1, which are incompatible with postnatal life. Everything is fine in utero, as far as anyone can tell. The baby is born, has to go on a ventilator, and can never come off. But of course, these infants are treated and treated and treated. Everything is done to try and save them. And it's very hard to know what the phenotype of their lungs actually is. So we could ask the question, if we now introduce the point mutations into our system, how do they differ from the expression, the wild type NKX2.1? So here you can see the wild type NKX2.1 makes very nice folded organoids with high levels of surfactant protein expression. And actually, what's key here is that surfactant is normally processed, normally trafficked, and secreted to the center of the lumen of the organoids. This point mutation in our assay has no effect, it's a very nice control, but clearly it does do something in vivo. But this particular set of point mutations in the homeodox domain that are truncations, and remind you that they're heterozygous in the infants, actually initiate type 2 epithelial cell development, but this stops, is incomplete. The cells remain quite columnar, and surfactant is inappropriately trafficked to the apical cell surface. So this is very exciting to us because surfactant is normally trafficked to the apical cell surface, endocytosed into multivesicular bodies, and then goes back into the secretion process via lamellar bodies. So we've now found essentially what we think what's going wrong in these infants. Surfactant can no longer be properly processed. And of course, we're very excited about further screening to see if this is a rescuable phenotype. So I'm going to stop here. Just mention again, of course, we're interested in the targets of NKX 2.1, which we've been exploring using the targeted DAM-ID approach. And just to summarize what I've been showing you, particularly about the alveolar formation. So using all these different genetic tools that we have now, we can also isolate cells from this alveolar formation stage of lung development. These progenitor cells self-organize very nicely and differentiate remarkably efficiently into both alveolar and airway cells. We've been using them to explore the signals, the transcription factors, and also key, I think, the cell-cell interactions that control this stage of lung development. And of course, having this very nice inducible type 2 cell differentiation is going to be a boon to doing various different, exploring various different respiratory conditions. So I'm going to stop here and acknowledge the people in my lab who really did this work, particularly Darway, who did all of the work I told you about developing the CRISPR tools and the screen, and Kunte, who's done all of the alveolar epithelial work, our various friends and collaborators, and thank you very much for listening and paying attention.
Thanks, Dr. Rollins, for your insightful talk on the lung organoids. And here's the question section, and I will take the privilege to ask the first question. So I just want to follow up on the end part of your story about the NKX 2.1. As you mentioned at the beginning, you're also interested in understanding the development mechanism of lung cancer, and we know that lots of NKX 2.1 has been studied in lung cancer. So I'm wondering the findings in lung organoids. Here you show how you think about its link to lung cancer.
I think there's a lot of work to do on NKX 2.1 still, is the short answer to that question. The data that's accumulating now suggests to me it has multiple binding partners in different settings. And actually, so I didn't say this, it's ubiquitously expressed in the lung. So it's in every single cell, and it's expressed at the same level as far as we can tell in every single cell, although it has these different effects in different cell types. So my current hypothesis is it's all about binding partners. Actually, loss of NKX 2.1, you start to get intestinal phenotypes in the lung. So lung and intestine are very closely related in development. And that's what happens in lung cancers. You lose NKX 2.1 and they take on a much more intestinal type phenotype. But the question is, which of its different binding partners is responsible for which of its phenotypes is going to be the key to answer.
Okay, but it seems you have the tools to identify binding factors. Okay.
Definitely. That's a big question for us now.
Hi, Emma. So I got one question. So one basic one is how SOX9 would control the stem cell maintenance besides the cell renewal?
I think that's what it does, essentially. It is controlling self-renewal. So it's promoting proliferation of the cells and it's inhibiting their differentiation. And it's controlling both sets of genes directly.
So it also have the critical function on the maintenance side as well, right?
Yes, definitely.
Okay. So the on the image that you show, that it seems to localize in the tip region, and then at that region actually the luminal polarity should also very tightly regulated by this SOX9. Any phenotype that you see the luminal polarity defects in the absence of the
no, this is a very good question. So all of the cells are polarized at this stage of lung development. So the lung develops from the foregut after polarization has already happened, and they're all polarized epithelial cells. So to be honest, we're a little bit unsure about this. At one point we thought there was, my student backed off it, was unwilling to put it in the paper. So possibly, I think, is a key answer to that SOX9 and polarization question.
Okay, so I got
But
one.
it doesn't
Thanks a lot for
directly-
your answer. Yeah.
Because I want to say it doesn't directly regulate any genes that affect polarization. So
Okay.
if it does, it's an indirect phenotype.
Okay, thanks a lot. So I would pick just one more from the audience. So and then we move to the panel discussion. So so we got this question that how these myofibroblasts around the alveolar tip express nocturne? Are they just a subpopulation?
So I think this is really interesting, and this is something we're exploring now. But actually, there's a transition. So the area epithelium, this is your airway, is surrounded by differentiating smooth muscle. Then at the stalk region of the lung, just adjacent to the tip, there's actually a myofibroblast population. So there's many questions here. Does the myofibroblast population give rise to smooth muscle, or are they independent? Is this the population that actually is growing out with the lung and then will give rise later to the alveolar fibroblasts? So we have multiple questions that we still need to answer, but they are a very specific subpopulation of cells that are uniquely here and uniquely express NOTAM.
Okay. Thanks a lot again, Dr. Rollins. So now, due to the time issue, so we move to the final part of our webinars, which is a panel discussion. And thank you once again for such inspiring talks from our amazing speakers today. So since we got a lot of questions from many in the audience that they would ask some general topic of about a organoid application. So the question that we can discuss for now, probably one is, are now the organoid ready for prime time, and any pros and cons using organoids? And for those very experienced using organoid for their research, it seems every lab, they would have their little niche for their own protocol. And what other elements or techniques can be integrated to those organoid application and benchmark the organoid findings? So we can start it from Mary, and then Hans and Emma answering those questions.
So if I understood your question correctly, you
Yeah.
were, at least the second part of the question, you were trying to ask us how much we can standardize the
Yes.
protocol.
Yeah.
To try to, I think that's a necessary step that the field has to do. So I think so far we've been very busy in developing systems that would work, but and of course everyone had their own recipe, or their own skill to do it in a particular way. But I think that now that many labs have demonstrated that primary tissue can grow, even iPS derived can grow, then yes, it's the moment to start standardizing. And I would kind of like to have a wish in that sense, that probably that we would have to do it in a kind of an international kind of consortium where the experts on the different organs set up what would be the standard, right? So in that sense, ISSCR recently is trying to put out guidelines on standardization of culture systems. I've participated on that. And I think that would be already the basis. But yes, in terms of what we add particularly to each media, we should develop standard protocols so everybody can benchmark to what has been done in Germany, to what has been done in China, and so on. But there will be, I can already foresee there will be kind of some maybe competition, because everyone will like to have their own protocol there. So we should go, the way I would approach the problem would be, we first set up standards for what we want to achieve, what function we want to have, which structure, which architecture we want to have, and then which is the protocol that fits best to that. Because at the end of the day, each culture might allow us to answer a particular question, and all is about, is your model enough to answer the question you want? That's my sense on it. I don't know, Hans or Emma.
Yeah, maybe you can add a few thoughts. Well, first of all, I think we should, you already showed this in the beginning, maybe, but we should really stress that all three of us have been talking about one of the two types of organoid technologies. So the other is the IPS or the ES-based organoids, and there are tissues that we cannot grow, and particularly the tissues that don't have a strong stem cell function, repair function, like brain or heart muscle. And that's, I think, where the IPS really are unique. And also our organoids tend to be strictly epithelial, everything. But if you think about epithelial cells in organs, they are usually the cells that perform this specific organal function, like in the lungs, as Emma showed, it's the airways, and the alveolar cells is where the air passes through and where we actually take up oxygen and release CO2. In the liver, the hepatocytes and cholangiocytes are the epithelial cells. But from our types, from our tissue stem cell derived organoids, we lack blood vessels, we lack stromal cells, we have to add them separately, we lack immune cells. And the IPS-based organoids tend to be more complete than ours. So there are definitely two different applications. If you study cancer, I don't think it makes sense to use an IPS technology, because there you straight go from a tumor to a cancer organoid, and then it wouldn't make sense to first grow an IPS cell from there. So that's one thing I wanted to remark. I fully agree with what Mary said. Of course, every lab wants to be unique. When you publish, you have to have something unique in your paper. And then when everybody has studied their protocol, and then to come up with a single protocol is going to be difficult in the way science works. But I think we really should do this. And one way would maybe to have the ICCR with their own journal, or with star protocols from Cell or so, really get per tissue type, get the key players together and see if we can agree on a single, or maybe a single basic organoid protocol, where then for different applications you can add different components. That should also be said. Yeah, and one last remark. So since 2 months, I'm actually I'm involved in a very large pharma company doing the research there, where I see that in drug development, organoids start slowly but steadily playing a role. And there it is extremely important that things are standardized, because otherwise they will never be allowed to use, or we, I should say now, will never be allowed to use human model systems to replace animal experiments, for instance, in toxicity safety studies, as models of human disease. I mean, I would strongly feel, and I guess Mary and Emma would agree, that these human model systems are probably better to model many of the human diseases that we all work on than animals can, depending on what the disease is. But then it's going to be key, but it's not going to be simple, and the ICCR might play a key role here.
Mm.
Okay, I'm just going to add one more point, because I totally agree with everything that Mary and Hans have just said. But I think the other element of variability is, of course, what cells you get at the end of the day. And I think everyone has a slightly different mixture of cell types at organoids. And it might be quite useful to tie into the standardization the human cell apps is trying to do on how cell types are annotated, as to also how we describe our organoids. That also might help us on this.
Chen, you have the another questions for the panel.
Yes, so I will ask a more open-ended question. So as all three of you have talked today, we know that in recent years, there is a big improvement of the organoid technology, and like we can make various type of organoids that from different tissues, and the utility has been greatly improved. And so my question is, in your opinion, what's the next breakthrough for organoids? For example, can we culture organoids from different organs together in the same system, or eventually can it replace organ? It's just an open-ended question. Thank you. Yeah, Mary, do you want? Sorry.
That's fine. I will just. I think there is a lot of. I think about it that we are still in organoids 1.0, and we need to move to 2.0, 3.0, and further. I think that the long final goal of many labs is to get a structure that recapitulates cellular interactions and architecture in the dish. Some of them are going to be very important to understand development. Some of them are going to be very important to understand physiology, and in humans specifically, where there are experiments we cannot do. It will also maybe be important for disease. It might depend the type of disease as well. I think this is, so adding complexity to the system, and by complexity, I mean vascularization, I mean additional cell types that we don't have. Pretty much what Hans was saying between the iPS-derived and the adult tissue-derived. It's true that the iPS-derived is much more complex, but still is immature, is a developmental type of culture. In the adult, I think the challenges in both are different. In the adult ones, I think adding complexity is going to be, I think, the challenge, but one of the things that many labs is going. But we should never forget that it's not about just putting cells together and crossing fingers. We should not aim for that. We just should really aim to recapitulate the tissue architecture and the tissue structure, because we can see different things. This is what I alluded a little bit on my talk. It's different when your cells are together than when you just separate them. To have total different outcomes. I think this is one of the directions, and maybe I pass on to Hans and Emma. So because I have other ideas, but I guess Hans and Emma, they also want to say something.
So I guess in Dutch, it's a Dutch saying, I don't want to cut the grass in front of Emma's shoes this time. So Emma, would you take this?
Happily, I'll be very brief. I think to me, it's the endothelium. And if we can add the endothelium to the adult organoids, this is going to open up an awful lot of different possibilities. I'm perfectly honest myself, less excited about combining organoids from different organs. This is what animal models are for. But I think adding endothelium will allow us to introduce an awful lot of complexity and ask an awful lot of different sets of questions. But for those iPS-derived organoids, it's all about the maturation. Also for my fetal-derived organoids, it's all about that maturation phase and what's post-maturation of cell types in the body and how we can do that in the dish. I think once we've got that, again, that will open up a whole new set of questions we can answer.
Yeah, so and I can add that I think what's going to be key in the next few years to integrate organoids better in various types of human disease modeling, drug development, is going to be combining organoids with elements of the immune system. They essentially are, because the human immune system doesn't translate to any animal, closest, of course, primates, but nobody's in favor of using primates for animal experimentation. It's getting to be harder and harder as well to do this. So immune systems, to model particularly chronic autoimmunity, for instance, which is essentially unresolved primary lung fibrosis would be another one. And the other is to incorporate better organoid models in infectious disease modeling. I think that turns out to be not so difficult for many, because often it's an interaction of one pathogen with one cell type that we can quite easily produce in organoids. And there's now multiple examples. You can do things that you cannot do in any other system using pathogens with, say, standard cancer cell lines, for instance, or in animals, where often the infectious agent doesn't infect, or when it infects, it doesn't give the right symptoms. Or often people use a related pathogen that does infect mice or cats or something like that, where you're just essentially studying a different disease. Yeah. That would be my result.
Thanks. So probably one more simple question is that it's that coming from the audience, that it's normally labor intensive and would need a very specified transgenic mice tool for those people that study stem cell lineage tracing experiment. So can we take an advantage of the organoid system to dissect stem cell sulfate decision for real time, or to identify novel stem cell marker? So whether and how that the organoid system can help people shorten the or giving people more advantage of doing a stem cell lineage tracing, or help to identify those critical markers for stem cell research.
Maybe I can start here briefly, because I'll have to run. So in the gut, where essentially we've been working for 12, 15 years now on gut organoids, I think we've really decoded all of the signals that will allow a stem cell to make each of these about 10 different cell types of the gut. It's a combination of Notch, Wind, EGF, BMP. Different combinations will give you different cell types. So you can almost make pure Paneth cells or pure enterocytes or pure goblet cells. And so I think the gut is a very hardwired differentiation system, maybe compared to others, but that shows that you can do, you can really resolve these signals in vitro. Having said that, I think what these organoids show is what tissues can do, but it doesn't mean that they actually will do this. So they give you all the possibilities, but I guess in the end you'll have to go back to an in vivo system to see what possibilities are used by nature.
No.
Okay.
And with that, I have to say goodbye because I have to run.
Sure.
See you all. Thanks to the audience for listening.
Thanks a lot,
Thanks for your time.
Dr. Hans-Clever. Yeah.
I would agree fully, actually, because every single time so yes, I think you can use organoids to detangle what happens in vivo, the example of lineage tracing you were saying, but actually we should not forget that it could still be an artifact. And we always have to benchmark. So until we have not benchmarked every single aspect of what we want to study, I don't think we can go and we can just say, oh, I got this finding of this cell as a stem cell in organoids. Now I know this is the stem cell in that organ. I think you have to go back and benchmark and actually that was exactly what happened to us when we tried to get out the liver, the first liver organoids in the mouse when I was still with Hans. We had to benchmark that the cell that we were saying that we could start our organoids and it was bipotent, that it was indeed in vivo. And it's very valid because at the end, the in vivo is the ground truth. But
I totally agree. I think it's really important, but I think the organoids are a really nice complement because I think what we can get from the organoids that it's a little bit harder to get from in vivo is that we can get this sort of real-time information and actually watch some of these cell fate decisions. So if you're confident that in vivo the cell fate decisions are really happening, then we have a very nice way of unpicking them in the dish.
Yeah, and then the manipulation is much more easier.
Yeah. Thanks a lot. Chan?
I think it's a time to wrap up.
Oh, okay. So now we are constrained by the time. So it's once again, we would like to thank the speakers giving us such wonderful talks and a very interesting panel discussion. And thank you all again for joining us today. We really appreciate your attendance. And once again, we also would like to thank Bing and Candice and the Westlake IT department to help us setting up this connection, and Chen Wang from South Stem Cell Press to organizing this webinar together. And thank you all again. And Chen will have further announcement for our next section.
Yes, we just want to take this opportunity to briefly introduce our next exciting webinar in next month, which is protein design and synthetic biology, which is supported by Cell Systems. And thank you again for your time. And we wish we could meet each other in person when in the near future.
Absolutely. Yeah.
Hopefully.
Okay.
Thanks for the invitation.
Thank you.
Bye. Bye, everyone.
Bye.
Goodbye.
Goodbye.